Okay, finishing up this chapter 3. I couldn't quite get the rest of prokaryotic in the first video capture, but I only need a couple of minutes to finish it. The rest of this is going to be eukaryotic and it's short. Okay, there's a little bit on transport. Remember we just finished the cell membrane in the earlier one? There's a little bit of transport that you should already know in this chapter. Remind yourself about passive transport and active transport, diffusion, osmosis, active transport mechanisms, ion pumps. Bacteria have similar things that you would have covered in general biology and A&P. A few transport things that you don't need to remember. Remember that you can have simple diffusion, for example. In the first scenario here, wait a minute, let me get back to that picture and let me get blue. Alright, so here you are looking at something like a gas going through a membrane, and you notice it just goes right through the membrane, needs no carrier. Then you've got something over here like maybe a sodium or potassium that goes right through a protein channel. Protein channel's in purple. Then in the third scenario, something like glucose that has to go through a cell. Notice that this protein carrier, it requires a protein carrier, but it's more than just a simple opening for the ion to go through like in the second scenario. In this case, the protein channel actually is involved with the ferrying or transport of the glucose through that membrane. But there is no ATP involved at all. You see no ATP, no energy involvement here. Then you've got water molecules on the far right. Okay, so these are all passive transport mechanisms. There's no picture of active transport here. But again, you've covered ion pumps before. Alright, so let me remind you about cell tonicity. Let's start with this first scenario here. Somewhere in general biology or A&P, you took pig blood and you put it into an isotonic solution, which was 0.9% NaCl, or if you want to actually be even more accurate, it's 0.85%, but we'll round it up to 0.9. When a cell, and let's say that this is a pig blood cell, it doesn't look like one, but make believe it is. You could have used epithelial cells from your mouth, it doesn't matter, would have done the same thing. You put any kind of cell into an isotonic solution, the solid concentrations inside the cell and outside of the cell are equal. In other words, the little dots of purple that you see out here, that tonicity is exactly the same as the tonicity inside of the cell. Therefore, even though water molecules diffuse through a membrane, you just saw that reference on the previous page, there will be no movement of more water molecules in one direction than the other. In other words, it's equal. You see the double arrow there. So these two solutions, inside and outside of the cell, are called isotonic. Now, if you do this to a plant cell, which is the scenario down here, same thing happens. By the way, cell walls do not get involved with transport. They are porous. Things go through walls. It's the membrane that's involved in transport. So in any case, you see a wall there in orange, and again, you see the solid concentration out here is equivalent to inside of the plant cell. They are isotonic solutions, just like in human cells and bacterial cells, and so on, and everything is fine. By the way, this could also be a bacterial cell down here, right? In fact, let's change it. Let's make it a bacterial cell, because they have walls too. Alright, now, let's go to the second scenario here. And notice that our cell now is in a solution where there's more solute out here look at the number of purple dots, compared to the solutes in the cell, less solute in the cell. So the term for that, wherever you have less solute, that solution is called hypotonic, and the solution with more solute is called hypertonic. And remember that water, when there is an imbalance in solution, you will get net osmosis. You'll get water moving more in one direction than the other. Water always osmoses towards the area of high solute, from where it's low to where it's high, in this scenario I've just given you, for example. Or you could say it a different way, water osmosis from an area where there's more water towards less water. Because in the area where you have less solute inside the cell, you're going to have more water. And out here where you have more solute, you're going to have less water. So water is moving down its own water gradient, water concentration gradient. But it's moving up the solute concentration gradient. Third, you can say it a third way, water osmoses from a hypotonic solution towards a hypertonic solution. So what's going to happen? You're going to get a net movement of water out of the cell shrinks. You saw that in red blood cells. You call that crenation. You see that there. Here's a bacterial cell in the same scenario, and again, look at the cell plasma membrane. It's this thing right here. And notice that it's pulling away from the wall. Alright, cell is shrinking. So these two scenarios show you dehydration of the cell. Okay, in the third scenario, look at this solution on the outside. There's less solute, I'm going to put S, out here. There is more solute in the cell. So now the inside of the cell is hyper, and the outside of the cell is hypo, and water's going to move. You're going to get net osmosis from hypo to hyper. The water moves in, you see that. The cell is going to explode. Now again, the top row pictures animal cells. You do this with pig blood cells. But look at the bottom here. Here's a bacterial cell in a hypotonic solution. And notice that the cell becomes very turgid. Compare that to, let me change to red, this scenario over here where the cell was in a isotonic solution. Notice the shape. There's the cell membrane right there. Look at the cell over here. It's much more plump. Now, it doesn't explode like an animal cell or cell without a wall, because the cell wall maintains the integrity of the cell. So that's what happens to cells when you put them in a hypotonic solution. You're going to get some death of cells. The longer they sit in it, the more damage there will be. But not all of them will be killed. So look down here, let me erase all that, at the bottom. You should be able to tell which of these scenarios represents on the outside solution, hypertonic, hypotonic, isotonic. On the far left, the two cells with a wall, like bacteria or plant, without a wall, like animal cells, they are in isotonic solutions. The guys on the right are in a hypotonic solution, and the guys in the middle are in a hypertonic solution. Now keep in mind this middle scenario, hypertonic. The cell dehydrates, right? Well, remember, at least 70% of a cell, or even more, depends on the cells, are water. So when you dehydrate a cell, it doesn't function well. And why is that important? Well, there are a lot of foods that purposefully create a hypertonic solution on the outside of the cells so that bacteria will not grow in the food. And this is done with sugar and salt. There are a lot of foods that are salt cured. Ham, bacon, some fish. Salt is probably the oldest preservative for food. Salt pork, you can buy that at the grocery store. Brine solutions. Brine solutions are high salt, and they use that to cure pickles, like dill pickles, things like that. Those are high in salt. Then sugar has been used as a solute to create hypertonicity, to dehydrate cells also for a long time. Let me think. Jellies, jams, honeys, syrups, all of those. So these are two solutes that are used for preserving food. The way they work is they create a hypertonic solution on the outside of the cells, if there are any bacteria in there. The bacteria dehydrate because of the water osmosing out towards the hypertonic solution. The cells dehydrate, don't have enough water volume, and they die. Okay, so finishing up with the prokaryotic cells. Inside of a bacterial cell, there's not a whole lot you find. I showed you one earlier. You find some ribosomes, so you see ribosomes down at the bottom of the list here. You have DNA floating around in there, but it's not in a nucleus. And then you've got some small molecules that are contained in little membrane-bound structures. They're called inclusions, cytoplasmic inclusions. I've listed a bunch of them here. There are more than this. They have varying functions. So, for example, let me point out some of these inclusions, most of the ones actually here, are for energy. Like a cell can use sulfur. If it can use sulfur instead of something like any kind of a molecule like glucose. A lot of bacterial cells would rather have a sulfur molecule to oxidize than a glucose molecule. Phosphates, starches and glycogen, things like that. These things are mainly involved with energy. The cell uses them as stored energy molecules. So down on the bottom right here, you've got a phase contrast micrograph of a bacterium that is storing sulfur, and you can see those little lit up yellowish things inside the cell. Those are sulfur granules. And the picture you're looking at right here are people, I don't know why they're wearing suits and ties, but anyway, they're in a sulfur pit, or on the edge of a sulfur pit, taking samples, and this is where you would find those bacteria that use sulfur. Then we've got some microorganisms that store lipids. These are lipid containing. They also can be used for energy. Excuse me. Then we've got some bacteria that can store enzymes in little packets. These are called carboxysomes, and they contain enzymes that can be used by some bacteria to fix carbon dioxide. Some bacteria contain gas containers that allow the bacteria to move up and down in a water column, allowing them to float higher, particularly to get to light, to sunlight for photosynthesis when they're in water. A lot of these bacteria may require, if they're going to photosynthesize, photosynthetic pigments like chlorophyll, in addition to some other pigments, and they contain them in inclusions. You're looking at a picture right here of one bacterium that does this. Notice that the water is red. This bacterium has a red pigment in addition to its chlorophyll that it uses for photosynthesis. Some bacteria, I just pointed to the wrong thing. There we go. Some bacteria have magnetosomes, which contain iron oxides, and they are used to facilitate the bacterium pointing to true north. So they act like little magnets in the cells. That's really about it. All right. Endospores. Look at the cell right here. Here's a bacterial cell. It's called a vegetative cell. It's not making a spore. It's your normal run-of-the-mill cell. And here it is on phase contrast microscopy. But inside, you notice that there is a thing forming right here called a spore. They're actually called endospores. When we get into fungi, you're going to hear about exospores. They form on the outside of fungi, some fungi, and they're not related at all to these. Eventually, you'll notice going down on this picture again that the spore is freed from the cell. The rest of the cell disintegrates. There is your mature endospore. And you can see them right here on a wet mount. This is phase contrast. These are unstained spores, and notice that the spores are forming inside the cells, and they're refractal. That is, they have different light-absorbing qualities than the vegetative part of the cell that's forming the spore, and it stands out at you. You can also stain spores. Here is a spore stain. We're going to be doing it in lab. You can see the spores are in green. Here is a gram stain of spores. This is a gram-positive rod, and you can see rods that don't have spores, but then of course you can see a spore right there and spores there. Notice that they don't stain. They don't take up the dye, the crystal violet, at all. Spores are extremely resistant to a variety of things. They're resistant to dye, resistant to heat, resistant to chemicals like antimicrobial chemicals and antibiotics. They are resistant to cold. They are resistant to radiation. They are totally dehydrated, dormant, last hundreds and hundreds and hundreds of years in a dormant state. When times get better, that is, if there's food, if there's rain, no radiation, the environment is better for them, they will germinate back to a vegetative cell. So they go back and forth between the vegetative cell and the endospore. The movement from, let's see, let me get rid of all this drawing. This change that you see right here from vegetative cell to spore is called sporulation. But then, remember, this cell dies, releasing the spore. The spore can live in a dormant stage, doing absolutely nothing, no metabolism, nothing, for a long, long time. But when times are better, the spore wall will start to become more pliable. Inside the spore, there will be a change in metabolic activity, hydration of the spore as the spore wall becomes more pliable and water goes in, and you start to get reading of genes, and this spore will now go back to the vegetative cell state, forming a cell around it, and the spore disappears. The spore basically disintegrates, but by that time it's already a regular vegetative cell, and that's called germination, going the other way. So the overall function of sporulation is survival against adverse conditions. There are lots of bacterial genera that make spores, but the two that you should know, that you want to know, because they are medically important, the genus Bacillus and the genus Clostridium. Bacillus contains the anthrax species, as well as a lot of other pathogenic species, and Clostridium contains Clostridium tetani, tetanus, Clostridium perfringens, gas gangrene, Clostridium botulinum, botulism. These are really big, high mortality diseases in humans. So most of the other spore-forming bacteria are not medically important. So here's a look, step by step, at what's going on inside of the vegetative cell up at the top. You can see that in the second frame, part of the cell is getting walled off, which will obviously become the spore. The spore wall has peptidoglycan in it, very high in protein. The spore is very, very dehydrated. It's got a lot of chemicals in it that are not found in normal, or at least in very large amounts inside the regular vegetative cells. Very high in calcium, high in dipicolinic acid. Notice that if you go about down to the 5th frame, the spore wall has become very thick, and then last, of course, the cell wall disintegrates, leaving only the spore. The pictures you're looking at the left, you can see a drawing with 4 spores, and this is just to show you inside the cells that spores can form at different places. So if you look over at the one on the left, it looks like a tennis racket. That's Clostridium tetani. It forms a terminal spore which looks like a tennis racket, which is very handy when you're trying to identify a species in a microscope. Some bacteria form central spores. Some bacteria form subterminal spores. So again, this is handy in diagnostics. And then you've got some electron micrographs showing spores forming inside of cells, and the one in the middle with green spores and a blue vegetative cell, just to show you the size and shape difference. Wanted to show you a really nice concept map. This is the website that you can see on the PowerPoint. So here's a look at just a few features of endospores, and there are the 2 important medical genera, Bacillus and Clostridium, that I mentioned, and just some key features, and again some of the diseases caused by these organisms. And this is a look at mouse lung. You can see the alveoli in yellow in the lung, and there's a bronchiole right there. It's been highlighted in blue. This is an electron micrograph. You see a blood capillary down towards the bottom, and there are spores that have been inhaled. So, other things inside of cells, we'll talk about nucleic acids much more later. Okay, so let's go to the eukaryotic cells and look at what they've got. You should remember all of this. There's not anything here that you shouldn't have ever seen before. So think about functions of all these organelles. Golgi for packaging of proteins. Vacuoles, which can contain a variety of things. They can contain energy for the cell, like starch. They can contain various gases, just like in prokaryotic. They can be excretory vacuoles, various things. Mitochondria for ATP generation, centrioles for mitosis and cell division, lysosomes for internal digestion, phagocytosis, nucleus obviously for DNA, chromosomes. ER, you got rough ER for remember that ribosomes attach to them, so they're involved in protein movement from where they're made at the ribosomes through the ER to other parts of the cell. You can see on the electron micrograph of the rough ER, looks like little channels in a cell cytoplasm, and the ribosomes are in black. Glycocalyx is just outer stuff on the outside of the membrane. Smooth ER for packaging, production of lipids, and so remind yourself about all that stuff. Let's see, I think there was... Nope, that's it. Okay. So external structures of eukaryotic cells. I took you through flagella and movement in prokaryotic, so here's movement in eukaryotic. You've already seen these before. Down at the bottom left, you see a paramecium here with lots of cilia, and these cilia beat, you can see that right there, and they propel the protozoan forward. And you see another picture of a ciliated protozoan over there, and here's a look at cilia embedded in the cell membrane. Then right above it, you've got an euglena with a single flagellum, which are much bigger, longer, wider diameter than cilia, and you see an example over on the right. Remember that these flagella of eukaryotic cells are very different than prokaryotic. Look at the arrangement right here. These are microtubules. You probably know this is a 9+2 arrangement. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, plus 2 in the center. That's the arrangement of microtubules forming a flagellum in a eukaryotic cell. In prokaryotic, you have no microtubules, so you don't have any kind of arrangement like this. Remember, there are no cilia in bacteria at all. That's only eukaryotic. Okay, just a couple things about cell membrane and wall. Let's go membrane first. There's really nothing I want to say about the membrane. It's the same phospholipid and protein bilayer that I just took you through. The one difference, 1-1-1 difference that I want to mention, cholesterol, steroids, lipids, cholesterol. You don't have that in prokaryotic membranes. Okay, so go up to the cell walls. You can have, that plants have cell walls. Algae have walls. In fact, the big multicellular algae are classified as plants. Fungi have cell walls. But they can have different things in their walls. So cellulose, which you already know is in plant cell walls. A lot of fungi have cellulose. They also have chitin. Chitin is the stuff that makes up the exoskeleton of shrimp and lobsters that you peel off of shrimp, for example, to eat them. And then in algae, there's a whole variety of different things that they can have in their walls. There is the origin of agar that we use in lab. Many of these things, by the way, are used by humans for different purposes. Agar, of course, is used in microbiology labs. Carrageenan, which is also a solidification material like agar, is good for adding to things like puddings. It's added as a thickening agent. And algin from another kind of algae is used by dentists to make molds for teeth. Like if you need a bite plane or something like that, they add a little bit of water to it. It's put into your mouth and it forms a hard mold, and then they make bite planes or false teeth or whatever from that mold. That's a solidification material, also can be found in cell walls. So there are at least 3 things that are very helpful for humans. Okay. Again, things inside of eukaryotic cells, I've already exposed you to that, so I will go on. At the end in your chapter, there's a nice comparison of prokaryotic and eukaryotic cells, what's present, what's not. It's a very extensive list, so here's a continuation, and that is about it for chapter 3. Oops, one more, sorry. I wondered why I didn't go there. All right, so I think this comes up actually in another chapter on classification. So I'm going to talk about this really in lecture. This is called the endosymbiotic theory, and it connects the evolution of eukaryotic cells to prokaryotic cells. It's called the endosymbiotic theory, and it basically, to summarize, says that the reason we have the way eukaryotic cells evolved with organelles, which you see on the picture, chloroplasts and mitochondria, is that way back when there were only prokaryotic cells, and there were prokaryotic cells of different sizes, some prokaryotic cells got into larger ones and became dependent on the larger prokaryotic cell. This is not a big leap to take, really, because there are predatory bacteria that actually can live inside of other bacteria. But in this case, these little bacteria started functioning for the benefit of their larger symbiotic host, like taking over functions of cell respiration and photosynthesis. The host cell, the larger prokaryotic cell, lost its ability to do its own functions with its little organelles, what would be called organelles, then taking over the job of those functions. As further evidence, remember that both mitochondria and chloroplasts have their own DNA. And so if in fact these were once prokaryotic cells that got inside of a larger cell, that would explain why there's DNA in there. So that's that in a nutshell. And a look at another variation of a picture of endosymbiosis. And another picture of endosymbiosis and the evolution of eukaryotic cells. That's it.